This tutorial will introduce you to the Structure-Function Linkage Database, or SFLD, which links enzyme sequence and structural information to specific functional information. The SFLD is no longer in active development, but the underlying data is still available in archive format and provides a useful example of how to organize protein information so that it can be efficiently leveraged to investigate sequence structure-function relationships. In this tutorial, I'll show you how the data in the SFLD archive is organized. In subsequent tutorials, we'll go into further detail on how you might use information downloaded from the SFLD archive. For example, to make hypotheses regarding the function of an uncharacterized enzyme. In this tutorial series, I'll be using the radical SAM superfamily, a diverse group of evolutionarily related enzymes that use a conserved 3-cysteine motif to bind an iron-sulfur cluster and activate S-adenosylmethionine, as an example. There are a couple of ways to get into the database. I'm going to start by browsing through the available superfamilies by clicking the Browse by Superfamily link at the top of the page. I get a list of the superfamilies in the database. Sequences in each SFLD superfamily are organized hierarchically based on sequence, structure, and function. At the top level of the hierarchy is an enzyme superfamily. The next level of the hierarchy is a subgroup. In the radical SAM superfamily, for example, we have 58 different subgroups, which have been largely defined based on overall sequence similarity, so that enzymes in each subgroup are more similar to each other than they are to other enzymes in the superfamily. Some subgroups are so large and diverse that they have been further divided into additional sub-subgroup levels. The next level down in the hierarchy is family, where, again, enzymes in a family have more sequence similarity to each other than to other sequences within the parent subgroup. But more importantly, they're thought to catalyze the same overall reaction based on the same mechanism. We currently have 98 families defined for radical SAM. The superfamily names in this table are clickable links. Clicking on any superfamily name will take you to the corresponding superfamily page. So I'll click on the radical SAM link to get to the radical SAM superfamily page. At the top of the superfamily page, we have some basic summary information about the superfamily. Because sequences sometimes contain multiple domains that may be different folds with different catalytic functions, we assign only the region of a sequence with the appropriate fold and function to a superfamily. We call this region a functional domain. So here we have listed the total number of functional domains in the superfamily. These are subdivided into those that have been assigned to a family and those with no family assignment. The functional domains that have been assigned to a family are further subdivided into those that share 100% of the family-specific catalytic residues identified by curators and those that don't, which may indicate that they're outliers. We also list the number of UniProt knowledge base identifiers and NCBI-GI numbers that correspond to those functional domains. And finally, we list the number of structures and reactions in the superfamily. Of course, because the SFLD is an archive, the underlying data is no longer being updated, so you should be aware that the counts listed in this table and in other places in the SFLD archive will likely be an underrepresentation of the current number of sequences, structures, or reactions. To the left of the summary table, you see an example superfamily structure. Under that, we have a brief text description of the superfamily. Clicking the References tab will give you a few literature references for further reading. The Cofactors tab shows you the cofactors used by all enzymes in the superfamily. And the Partial Reaction tab shows you the chemical capability shared by radical SAM superfamily members. The Download Archive Data tab lets you download various data relevant to the superfamily. There are several types of files available for download here, so take advantage of the text in the description column to find the file you want. Files available generally include a FASTA format file containing amino acid sequences for all proteins in the superfamily. You'll also find a tab-delimited annotation file containing annotation information for all sequences in the superfamily. This annotation file contains things like subgroup and family classification, species, PDB identifiers, and much more. You'll also find a Stockholm format alignment file containing a curated alignment of a representative set of sequences from the group, with important catalytic residues annotated. You'll also find several different types of network files in XGMML format, which can be opened with the Cytoscape program. The representative network file is a sequence similarity network, while the other files are various different types of chemical similarity networks. The one described as molecule network, all, contains all substrates and products from all reactions catalyzed by enzymes within the superfamily. The one labeled molecule network, product, contains all products from all reactions catalyzed by enzymes within the superfamily. The one labeled molecule network, substrate, contains all substrates from all reactions catalyzed by enzymes within the superfamily. And the one labeled reaction network contains all reactions catalyzed by enzymes within the superfamily. For more information on the networks available in the SFLD, you can refer to the Networks page. You'll find that by scrolling to the top and then clicking on the Documentation menu. And there's an option for Networks right here. You can also refer to parts 2 and 3 of this tutorial series for more information on how to use the networks available in the SFLD. Finally, for superfamilies for which a recent paper has been published by the Babbitt Lab, you may also find a download option for networks and other data specific to that paper. One last note. Some of these files, for example, the FASTA format sequence files and chemical similarity networks, are only available for download at the superfamily level. Other types of files, such as sequence similarity networks and alignment files, are also available at the subgroup and family level. So if you wanted a sequence similarity network of a particular subgroup, you could download that directly from the Download Archive Data tab of the relevant subgroup page. At the bottom of the superfamily page, we have a tabular view of the way the sequences and structures are distributed between subgroups and families. Subgroups are shown with a blue background color, and families are shown with a yellow background color. The T, K, C, U, and S column headings have mouse-over explanations that describe the data listed under each. And all of the subgroup and family names are clickable links, so if you wanted more detail about a particular group, you could click on the associated link. That's a very brief introduction to the SFLD archive. If you'd like to explore how to use the data from the archive in more detail, see parts 2 and 3 of this tutorial series, where we'll explore how to use SFLD networks to make hypotheses about the function of an uncharacterized enzyme.